So now I'd like to talk about another set of techniques which are known as electrophysiological techniques or neuroelectrophysiological techniques, and they rely on the electrical activity of neurons. So the fact that your neurons actually utilize action potentials to send messages along their lengths is significant in terms of development of these techniques, because you can actually disrupt the electrical transmission in a particular region, and then see what happens. You get a sense of what the functional, aspect of that particular region might be, or that particular pathway might be. And you can also measure the activity of a particular region. As, for example, we discussed in terms of identifying what different columns in primary visual cortex respond to. Out here in the visual world, those bars of light would set off electrical activity in particular columns and not in others, right? So I just want to give you a little bit of the history of this. So Charles Darwin wrote a book on the expression of emotions in man and animals back in 1872, when he used a lot of these images from this one researcher who actually was interested in the neural architecture, the electrical architecture underlying facial expression. And so this is actually a wire here, or an electrode that's actually releasing electricity and stimulating some branches of the facial nerve, which moves the muscles of facial expression. And this was actually a relatively painful way to examine the underlying sort of electrical architecture of facial expression, because you had to pass through the skin, and it probably wasn't all that pleasant. But you could get, by stimulating various branches of the facial nerve, you could get different muscle groups. There are about 44 muscles underlying the expression. You get them to sort of have coordinated sort of output and activity. You could explore that underlying kind of electrical architecture of facial expression. Now, a researcher, a neurologist actually, who did a great deal of research on the brain in terms of stimulating it directly with electrodes, is somebody named Wilder Penfield, who was, again, a neurologist at the Montreal Neurological Institute in the 1950s. And he basically helped to confirm the existence of what we refer to as those motor and sensory maps, right, in frontal lobe and parietal lobe that we've referred to before. The development of the existence of what's called the homunculus, right? And a homunculus, just refers to a little creature that you can sort of generate based upon the degree of representation of different body parts in the brain. So, for example, Penfield would take patients, actually, who were typically undergoing surgery for epilepsy, for intractable epilepsy, so epilepsy that couldn't be treated with, successfully with drugs. And the decision had been made to actually go in and to remove areas of brain tissue that were damaged by repeated seizure activity. And some of this tissue was clearly damaged. You could visibly see the damage. There was epileptiform damage, but other areas nearby could still be the source of additional seizure activity. There may be something wrong with the wiring, but however, it wasn't visibly damaged the way other areas that had been, consistently involved in seizure activity were. So Penfield would take an electrode and he would literally apply it directly to the surface of the cortex, and he would, have his patients who were awake during this procedure, carry out various tasks and see if stimulation in this region would disrupt those tasks, or if they reported anything unusual. And he found that when he stimulated the precentral gyrus, remember this is the primary motor strip in the frontal lobe, right in front of the central sulcus, he found he could get body parts on the opposite side of the body, if he were doing it over here, to move, right? And there was a mapping of the opposite side of the body, just basically present on the precentral gyrus, or which is now known as primary motor cortex, or the motor strip. Work since Penfield's time has shown that if you actually deliver a longer pulse of electrical activity, which is actually more physiologically relevant, probably, like more like about 500 milliseconds. This is work done by a man named Georgopoulos up at Minnesota, they would find that they could get more complicated sorts of responses. So, for example, if they went over the hand region of the homunculus, they might get the hand to go like this and move something to the mouth, which is interesting if you look at the way the homunculus is kind of arranged here. It's not a one-to-one mapping, certainly, of course. You got the big hands, the big mouth, the big, tongue, the big larynx, pharynx, and the small torso. That reflects, of course, the degree of fine motor control. And over in the postcentral gyrus, in the parietal lobe, it represents the degree of sensory, detection ability. But what you should also notice about this homuncular mapping that Penfield identified is how it's not a continuous mapping. I mean, the hand suddenly jumps up to the face. Well, why would that be so? Well, you're doing this an awful lot, or you're bringing things to your mouth. So it may have another kind of organizational, reason for its specific organization. Penfield also found that when he stimulated, for example, if he stimulated, short bursts in the precentral or postcentral gyrus, he got reliable reports of, he got reliable movement of particular body parts on the opposite side of the body, and reliable sensations from those body parts on the opposite side if he were stimulating in the postcentral gyrus or somatosensory strip, which is known as S1. However, if he stimulated in other areas, he found he got less consistent reports, but sometimes he could generate, a powerful memory recall in somebody, and these were some of the areas that he found would generate a powerful memory. It wasn't always a consistently reported memory. If you stimulated again, he wouldn't necessarily get the same response. A more recent paper, actually, it's not super recent, but a very well-known paper, actually, by Fried et al., actually reported on a patient who was undergoing surgery for epilepsy, and this was focused in her left frontal lobe, and they stimulated actually a portion of the left frontal lobe. It was near the supplementary motor area, and this particular patient just started laughing, and she had a strong sense of merriment and enjoyment, and she would attribute it to whatever was present in the area at the time, or whatever was happening at the time. So I'm going to, just to read this from, this is the abstract for this particular paper, but she said, they write, the patient attributed her laughter to a variety of external stimuli that were presented during a sequence of tasks, such as naming objects, counting, and finger-to-thumb apposition, none of which are thought normally to be intrinsically funny. So, this is obviously like a higher level region that attributes, enjoyment or humor to a particular situation. And it's probably a place you'd want to stimulate on a regular basis. I should mention there is another technique that is utilized, another sort of electrophysiological technique that employs magnetism.